Here at AACR, we highlight the work in colorectal cancer research. We have Alan with us from the University of Connecticut. Alan, tell me about your poster.
Hi, we're interested in ACS, which are putative precursors to colorectal cancers. These are pre-polyp stage, less than 5 millimeters in diameter.
And ACF stands for?
Aberrant crypt foci. We were interested in looking at the transcriptional consequences of these early lesions that actually harbor these very early mutations in KRAS, BRAF, or APC.
And it's very early.
Yes.
We're talking about these, the vast majority of these have no consequence whatsoever.
Yes, it's kind of highly debated whether or not they actually have any clinical relevance. We believe a subset of these actually progress to colorectal cancer. And we wanted to identify the pathways that might be involved in actually kind of mediating the progression or the regression of these lesions.
And then how did you take a look at that?
So we used a combination of LCM, laser captured microdissection, and AmpliSeq, which is a targeted RNA-Seq technology, to actually look at the transcriptional profile in both the epithelium and also the stroma of these samples.
And I remember from your poster, you were looking at either senescence or apoptosis,
Yes.
Is that correct?
So I've mentioned before, we were interested in the progression or regression, and pathways that are involved in actually preventing these could also be senescence, which actually may inhibit the progression of these lesions, or apoptosis, which might cause a regression of these lesions as well.
So you're able to separate the ACF from the stroma.
Yes.
And then what did you do?
So we were able to actually find the transcriptome and use principal component analysis to show that they actually cluster differently. Epithelial samples clustered from stroma samples, and within those two tissue compartments, the precancerous lesions separated from the normal samples.
So you had AmpliSeq RNA to do targeted RNA-seq.
Yes.
And the gene sets were involved with senescence or apoptosis, is that correct?
So we used the off-the-shelf 267 apoptotic gene panel that existed already, and we also made a custom 21 gene panel with relevant senescent genes.
Yes, I see. And then you showed some significant differences then.
Yes, we were able to find mutation-specific changes in the epithelial tissue, as well as sample-specific changes between normal and ACF. Some of the highlights to that were we were able to find immune cell population changes and changes to the phenotype of the fibroblast of the underlying stroma.
So the leukocytes that were infiltrating,
Yes.
then, you found these signatures of leukocytes
Yes.
inside that stromal tissue.
Yeah, so in the apoptotic panel, there were two genes that were only transcribed by lymphocytes, and we found those to be highly upregulated in the stromal population.
Really interesting.
Yes.
Now, you wouldn't be able to find that unless you separate out these particular cell types from the ACF versus the stroma, is that correct?
Not exactly.
Not exactly, okay.
But we were able to show that in the stroma population, at least, that you could show that the gene expression in the stroma was higher, but you would not be able to tell if you looked at the entire tissue.
Right. And then why didn't you just use total RNA-seq?
So the advantage of using target RNA-seq is that we're only looking at a very small subset of genes, and we're able to have a very efficient, both on a cost perspective and also a sample perspective, the read density required to actually get enough reads to actually do adequate gene expression profiling.
That's great.
Yeah.
Well, thank you for taking the time to speak with us today.
It was a pleasure talking to you.